precision effect variegation very well. And I want to only tell you about one that was hugely important in the field of epigenetics. Let me ask you to look at the bottom of this box where you see something called SUBAR39. That means it's a mutation that suppresses this phenomenon I've been describing, precision effect variegation. And what's the SUBAR39 part all about? That stands for, it was a mutation that was mapped on chromosome 3, no big deal, and it was mutant number 9. Okay, chromosome 3, mutant number 9. And of course, the important part is, what did it do? What was its role? What was its molecular properties? And I get to tell you one of my most favorite sort of stories in my career was when the individual shown here in the lower right, Thomas Genuine, literally obtained some evidence that the mouse homolog of SUBAR39, not the Drosophila, but the mouse, smelled like it was a methyltransferase writer. And he wanted to team up with a lab like mine and see if we could figure out if that was so. And so using a lot of the same sort of approaches that Jim Burnell and we had done with several writers, we teamed up with Thomas's lab, and shown here is the blackboard from my office to this day, down the street. And the biochemical take-home message was that the mammalian SUBAR39 was indeed a methyltransferase. That means it's a methyl writer. And remarkably, its substrate, its target, is H3, histone 3, at what position? Lysine 9. Unbelievable. So this SUBAR39 was named correctly all along. And I think Thomas and I still, we're very good friends. We laugh about this. You know, it was just unbelievable that the geneticist named this right all along without even knowing what it did. So what's important? I'll give you sort of now sort of a raging insight into what people now think. On the left side, it looks like there are situations where you want to activate genes. You might do that by taking these acetyl groups, enzymatically adding them with a histone acetyltransferase writer. Now we know that once that mark has been written, there are proteins that read that mark, what's shown here, protein X. They'll dock on that acetyl group. They'll bring about gene activation. And when you really want to shut that gene off, all you have to do is employ those HDACs that will strip that acetyl group off. And so that's a very sort of attractive, well-worked-out paradigm in the field. In contrast, let's take a situation where you want to silence the gene epigenetically. No mutations, just silence the gene epigenetically. Well, in this case, you're going to have to make sure that lysine residue, that's the blue K in these slides, that's the one-letter code for lysine, you're going to have to make sure that you don't have an acetyl group there. So you might employ the HDAC to remove the acetyl group as step one. Then you'll go to the histone methyltransferase shown in red, and you'll add a methyl group. So that's this HMT, that's the methyltransferase writer. You might think of that as the SUBAR39. And then once that methyl mark has been put on, there'll be readers, like shown here in Y, in red, that literally silence and compact that chromatin by reading the methyl mark. And I have to say, it's funny to me that when this slide was made, I thought it was pretty nice, but we didn't know, and I don't show on this cartoon, was there a demethylase? Was there an enzyme that took the methyl groups off? And now indeed, Yangshi and his colleagues, and now many others, have found that even these methyl marks can be enzymatically removed. So in this sense, this slide is even out of date. But nonetheless, acetyl marks generally track onness, and methyl groups generally track offness in chromatin. So a couple biology examples. Here's another nice cartoon. It wasn't drawn by someone in my lab. You can see it's mom and dad doing their thing, but the sister's clearly irritating the brother by saying her epigenome. Now, I put in the epi. Her epigenome is more complex than his. And I think that this is a case where she's right, because every woman has two X chromosomes. So that's underneath her, the 2 pink chromosomes, two X chromosomes, where every male, young boy, father, whatever, XY. So, how does nature create a balance on those X chromosomes? There's 2 in the female, there's 1 in the male, and what they've learned to do is to literally shut off early in development one of those X chromosomes. That's called X inactivation. And you'll think I'm making this up, but when you take female mammalian cells and stain them, there's an example of 2. You can't see much staining on the left panel, but if you stain with a methyl antibody, that's a methyl histone antibody, I hope you can see there's little patches that light up red. And those are the completely shut down heterochromatic inactive X chromosome. If you did the same staining experiment in a male cell line, you won't see any of that. And as a footnote, if ever you've seen a calico cat shown on the right, that coat color patches is all a reflection of female X inactivation in the cat that happens to be tracking to a coat color gene, and that happens early in development, and it makes different coat colors and different patches, hence the calico coat properties, and calico cats are always female. They're female because of this coat color phenomenon that tracks back to X inactivation and methylation. Identical twins, I mentioned, we should know that they are absolutely identical genetically, but I think if any of you know identical twins, they're often not identical. It is becoming an attractive model system, not these 2 individuals, where people have studied different behavioral properties, different disease properties from monozygotic twins, and found out that they're not identical with respect to their epigenome, and that now it becomes a nice model system for identical genetics, but not identical epigenetics. And then, of course, I certainly grew up thinking that if I wanted to be abusive to myself, that's not so cool, but at least I'm only hurting myself, so it was a life choice that I was making. And I think a lot of people might have said, we are what we eat. But much more recently, because of this revolution in epigenetics, others have written other alterations of that quote that might be, we are what our parents ate, and maybe our parents before them. And I'll give you a nice visual of that. These are identically genetic mice. But the only thing that was varied in this experiment by these researchers was the diet that their mother was given when these babies were in utero. So the question was, wow, I just altered the diet of the mother in too much detail. These were alterations in a diet that would foster the methyl modification, and you can see that the different pups, all genetically identical, are phenotypically very different sizes, very different coat colors, and that's kind of a scary thought. They're identically genetic in terms of genetic terms, but the diet of the mother impacted the outcome of the children. And if you take this to one more extreme level, Michael Meany and his colleagues have been asking questions that fall more in the realm of social behavior, and that would be, what about having a model system that might mimic good moms versus bad moms? And he's got mice that actually are very, very good moms, meaning that they lick their pups a lot, they kind of hold and cuddle their babies quite a bit, and he's found some strains of mice that are just the opposite. So we'll sort of call them bad moms. They don't cuddle, they don't lick, and he's then asked, what happens if I can expose genetically identical mice pups to good moms or bad moms? And as you might guess, he's now paying a lot of attention to epigenetic marks, and he's finding out that marks change in the brain regions that he studies that are responsive to ultimately alterations in gene expression that lead to alterations in stress and inherited phenotypes. And it's quite amazing to then think that this may be socially relevant in our society when we have unfortunate situations that track good moms versus not so good moms, not only in terms of the tragedy of it all, but also the upbringing of children. And perhaps can traits be passed on to even their children that are inherited through this epigenome layer. So this would be a summary slide. I think you've seen it before. I want to leave you with the impression that on the left side is the classic textbook genetics. There's nothing wrong with that. And in fact, it's very clear that some things that are like smoking and too much sunlight, they can indeed alter the DNA mutations that can cause disease. And that's certainly something we do want to protect and hopefully inherit good copies from mom and dad. But there is now an emerging, if you will, revolution in an understanding that genetics doesn't explain everything. And there seems to be, in hindsight, you might think, a much more dynamic opportunity for these genomes to be responsive much more quickly in time to needs that might be epigenetic in nature. I should mention for completeness, it's not just the histones that are chemically modified, the DNA as well. And these things are now a raging topic for biochemists like myself. Again, writers, erasers, readers. And what's really rapidly ramps up the interest in these modifications is that many, if not all of them, are now associated with human diseases, notably cancer, and many are being targeted for drug development. Why? Again, because the genome is relatively unfixable. The epigenome is much more responsive to being fixed. So if you're interested in this topic, I'm not trying to plug this book, but a pleasurable sort of challenge for it. It's a complicated field, so some time ago, Cold Spring Harbor approached a small number of us to write a textbook on the subject. And if you're interested, this is the second edition of a textbook. It was published, came out this last year, and importantly, it makes me sort of want to introduce you to the editorial team. You might recognize Thomas Genuine on the far left, because he's actually the scientist that we enjoyed working with on the methylation and the Suvar 39. That's been along the friendship, and Monica Lochner, Danny Weinberg, myself, and Barry Lord Kaporas are all part of the editorial team, which helps me make the point that besides your lab mates in the lab, it's equally a privilege and fun to sort of carry out worldwide interactions and collaborations, and in this case an academic project with so many talented people. So I'm done with my first lecture. I would be remiss if I didn't thank many people. Of course, anything I referred to, I showed you a few lab people's pictures like super now, but I've had a rich career of many talented students and postdocs and technicians. We don't do many of our projects without many important collaborators. The iBiology staff here has been wonderful. I'm not used to being filmed or any of these productions, but they've treated me really nicely. The Lasker Foundation for allowing this to happen here, and my funding sources, Rockefeller University, and you all for your time and attention. Thank you very much. いう形でございます。ですから、このルミノっていうんですが、これ、小中毒でありますが、これを使うと、低濃度の二酸化炭素と反応して、結果的には、ずっと 5% ぐらいまで、ほぼ同じような結果で、それ以下はちょっと正直やってないんですけども、おそらくもうちょっとしかできるだろうと思います。これを出荷で出して、還元が可能である。加圧水素はもちろんございません。で、リーターという方法によって、二酸化炭素が選択的に、この場合、二酸とか、メタノール、メタナンといいます、CO だけが選択的に集中いたします。で、CO というのはご存知のように、水素と反応させれば、酸化水素原料になります。ということで、有害な反応ではないかと考えております。で、じゃあ、原料を使わないといけないかというと、そんなことはありません。今、我々のしてからマンガン、という意味でも同様の反応があります。特にマンガンだけの戦略的な意味があるのかもしれません。で、この場合も低濃度の二酸化炭素と反応して、同様の付加体を作ります。CO2 の付加体を作ります。で、この場合はですね、マンガンとルテニウムの場合は、生成ですよ。これ、微酸になります。塩でなくて微酸になります。ですから、反応ではほとんど一緒なんですけども、非常に作用を変えることによって、塩と塩するか、塩と微酸にするかという選択ができます。で、微酸がどれぐらいがあったら、今、微酸に関しては、そこから投入すると、そこから水素が出ると。水素のCO2に変わるという反応がよく研究されています。ですから、水素で微酸の液体ですから、水素の貯蔵運搬に適した純体として期待できるんじゃないかというふうに考えております。ですから、こういう金属なんですけど、まだ他にも可能性があるのがあって、今、我々チャレンジしているところでございます。ルートです。これは私がどのような話がなくて皆さんがよく考えられることではないかと思うんですけれども、一つ考えられるのは、やはり排出量から出たやつを濃縮するエネルギーを生じ方の濃縮プロセスをどういった形で二酸や二酸化炭素に変換する技術ですから、そういう形をおそらく再生可能エネルギーを使ってやるということになるだろうというふうに思います。二酸化炭素の場合は酸化水素に変換できまして、二酸は水素のペレになるということになるのではないかと思います。で、これは技術が分かる技術ですので、どういうふうに使って、これからどこまでやられるかかなりたくさんの研究が必要です。ですから、特に我々はもともと光カーブの専門家ですので、電気カーブに関する深い知識をお持ちの企業の方に協力していただくと、進展する可能性があるんじゃないかと期待しております。あと実際どういうものがあるんですね、多量子標とするというのはどういう可能性のようなものなのかということも、重要なことなんだろうというふうに思います。時代関係はこの回から総理で、常識実際の方から出馬させていただいております。で、運送のことはこの後私に聞いていただければいいんですが、全部に関することは、独立業者法人工業所有情報研修機関の辰巳の方に聞いていただければというふうに思います。それのところの時に関しては支援していただきます。はい、以上です。ご紹介ありがとうございます。山田大学の宮崎と申します。本日はこのような人柄にお招きいただきまして、GSP の関係者の方々、そして私プレスはいわゆるエネルギー境界面という領域で研究をやらせていただいておりますけれども、その領域の方々に厚くお礼申し上げたいと思います。私の本日ご紹介させていただきたい研究の内容につきましては、高性能なアニオン等電性発光ということで、大体ですね、膜の厚さが10ミクロンから20ミクロンという非常に薄いプラスチックの膜ではございますけれども、非常に高いですね、イオン導電性をと、そして安定性を示すということで、このクエストの領域の中では、特にですね、アルカリ型燃料電というものに焦点を絞って展開しておりますけれども、その中でですね、この性能というものが必ずしもその燃料電池だけではなくて、他のエネルギーデバイスにもどうも展開できそうだというような技術だというふうになってまいりましたので、本日お越しの皆様方にその内容をご紹介して、ぜひそのある方々には詳細なお話を進めさせていただきたいというふうに考えております。で、今申し上げましたこの電動電池というものにつきましては、すでに皆様ご承知かと思いますけれども、燃料電池の自動車、あるいは家庭用の燃料電池という形で実用化されて、徐々に我々の生活社会の中に入ってきているという段階でございます。で、その燃料電池につきましては、現状ですね、ほとんどのものがこちらの左側の図に示しましたように、そのイオンとしてはですね、プラス特に水素のイオンが連動するような膜を使っているということでございます。その理由は、このプロトンというものがイオンの中で最も小さいと